Hello, anatomy and physiology. We are here today talking about blood types. And so we will take a little bit of notes in your packet, but quite a few of the first few slides we're going to go through. There's nothing to write down, mostly just information, so that hopefully this stuff will make sense to you. First off, you should have done before watching this video the blood type simulation lab activity. And I recorded the pre-lab and then the results of the post-lab. And so this should already be done and in your lab notebook. So if you haven't done it already, it's a good thing to do before you watch this video. If we were in class, I would have you do this lab before we discussed blood types because it shows you a visual representation of whose blood is safe to mix with whose and whose isn't, using food coloring, red, blue, purple, and then just water. You also should have gathered the information about who is the universal recipient, the universal donor, and who has the most common and least common blood type. And just to write that down, there's no conclusion or statement of significance or even a hypothesis necessary on this. It's just the question and then the observations. What blood is safe, which one's not? Just a little shorty for you to do. And there's an option for you to do it at home if you're bored and want to do some science. But this is a picture, just because the video wasn't as clear of the pre. In fact, this picture is a little bit better. Before transfusion and after transfusion, so you can kind of compare the differences between them. The ones that stayed the same color as they were before are considered safe, and then the ones like this that have changed color are considered unsafe. The purple doesn't mix awesome at times, and so all of the ABs are considered safe. And if you take a look here, the O's were all clear, just plain water. And when you look at it over here, the O's changed in every single one except for this last one where O mixed with O. And so that should give you an idea of who the universal recipient is and who is the universal donor, because everyone else is in that column stayed the same. And so on those few color changes that are a little hard to identify, I've provided for you at least those safe and unsafe ones. This is safe underneath here, just in case you didn't get that written down. But again, if you haven't yet done the lab simulation, you should do that before watching this video. Alrighty. So what are blood types? We have markers on the outside of our red blood cells and all of our cells, but specifically our red blood cells have specific markers that say if they are A, AB, B, or O. And this image and the next few little cartoon images should look familiar. They're from the video I asked you to watch. I think last time we had class. Those videos are important and helpful, the supplemental things in addition to these lectures. There's something I would show in class if we were present in class and have you watch it as part of the material. So if you are A blood type, it just means that you have these markers, these little blue dots here saying that you are A. If you are B, you got these little yellow spikes on them saying that this is my blood type B. It's a marker on the outside. AB has a mixture of these two, and O has none, neither A or B. So what those markers are is actually a throwback to our tissues and cells chapter. So this picture should look familiar from a previous packet, not their current one, but we labeled the outside of our cell membrane. The thing that I want to pull attention to here is the glycoprotein. And this is, we talked about, and I described as being your jersey, right? This is the marker that identifies your cells as belonging to you. So Mrs. Lloyd has markers inside every single one of her cells that says Team Lloyd, and they're all cells that belong in my body. The same is true of my red blood cells as well. Now, that marker specifically on our red blood cells, we call those antigens. And I like to remember the difference between an antigen and an antibody is it's got this big I in it. The antigen with that I think of it as a nice big capital I. That's me, I, my blood. That's my type, my markers. It belongs to me. So think of that antigen, that big I, that's mine in there. And so when we look at the markers on the outside of our red blood cells, they're just glycoproteins, same thing that we've talked about before. So when we have markers on our cells, we have markers for the things that don't belong. And we have lots of antibodies in our body. If you've been vaccinated or ever have had chicken pox, you should have antibodies for the chicken pox virus in your body. And they just float around, okay? They're just floating around in the plasma, looking to see if some puzzle piece matches with it so it can mark it. So if, let's say, I have antibodies for a bacteria, this antibody will attach to it and mark it for destruction. And so these antibodies, they are anti someone else's body. I don't want their body in my body, so I go and fight them. And so we have this little cartoon here that's a white blood cell slicing, killing this bacteria because my antibodies, I don't want no one's body in my body. Okay, I'm going to go ahead and attack it, mark it, so these guys can go and fight it. The same thing happens if I get the wrong type of blood in my body. So this is me. Well, actually not really because I'm O, but this is A blood type. If somebody was donated the wrong blood type, what would happen is they have antibodies to fight it, the wrong blood type. And so these antibodies would adhere to the antigen on the foreign cell, causing it to clump up. And that term we use for clumping is called agglutination. Agglutination. It's very similar to coagulation, except we use the term agglutination when we're talking about red blood cells from transfusions clumping up. Agglutination, but it's just red blood cells sticking to each other because of these antibodies. Again, not my body. Antibodies are going to mark the enemy, basically. Okay, they aren't wearing my team jersey, so we're going to mark them and fight them. All right, so again, as I mentioned, these are our 4 different blood types. AB has both markers. A has its own, B has its own, and O, which is what Mrs. Lloyd has none. Now, from the activity, the lab simulation, hopefully you could discern who the universal recipient and universal donors were. So what makes AB interesting is that we usually have antibodies for the antigens that we don't have. So, for example, if I have, I think this one's A blood, we have B blood. In fact, I'll go back here. Right, so this is A blood. These are B antigens. This is B blood, and so those B antigens fought and attacked this one. I don't have any A antibodies in here because it would attack my own cells, and I don't want that to happen. So instead, we would have different antibodies that do not match our outside of our cell. So in the case of AB blood, they have no antibodies because they have both types of antigens. Remember, antigen I, that's me. Because it has both types, it's wearing like 2 jerseys, then there are no markers to attack it. So this is why it's the universal recipient, because it can take anybody's blood and it won't attack it. It won't attack the other blood. So in the case of the universal donor, which is O blood type, they can give their blood to anybody because there's no markers on it. So it doesn't matter what antibodies the receiver has, they wouldn't attack O because there's nothing to attack. There's nothing to adhere to. The problem with having O blood type is that you have both types of antibodies. So if you get A blood, you attack it. If you get B blood, you attack it. And if you get AB blood, your body attacks it. And so O's, although they can give to everybody, they can only receive also from O's. And so we're going to fill out a chart that hopefully will clarify a lot of this. But O is the universal donor, AB is the universal recipient. The other piece to blood type is if you are positive or negative, and that has to do with your Rh factor, which comes from the rhesus monkey, which it was discovered in. Rh positive just means that they have markers, they have the antigens on their body for the Rh factor, which is really, they call it a D antigen. But because they have the antigen, they don't have any antibodies. So much like AB blood, they won't attack whether they get O blood, or sorry, negative blood or positive blood. They can accept either. But if an individual is Rh negative and they get positive blood, the same thing, agglutination would happen and their cells would stick to it. If it's the second time they've come in contact with blood, and positive blood, and I'll explain that here in a bit. All right, so this isn't in your packet, but we're getting close before you're going to start writing some stuff down. But I do like this graphic because you can see the markers on here. So each of these are the red blood cells. Okay, we have them labeled A, B, and O. And each of them has the antigens. Remember, antigen belongs to me. It's my team jersey. And so A is wearing the A jersey. Okay, you can see all those studded around. B is wearing the B jersey, antigen. AB is wearing both. And O is streaking. Okay, it doesn't have either. It's not wearing any on there. But the antibodies that are present are opposite of the antigen. Again, the jersey. So notice how the B antibody would fix and adhere. It's got kind of that same little shape here as the B's would. And so it would attack a B cell, or it would attack an AB cell, because this would match up with this. If you have B blood, you have the anti-A antibody. Notice the little curve shape. So it's going to adhere to anything with the A antigen. So that would be either A or AB. But no one would attack O because, again, no jersey, nothing for it to adhere to. AB, because it's wearing both jerseys, it has none of the antibodies because it would be fighting itself constantly. And then O, notice, has both on its cells as well. Okay, so now we're ready to actually fill something out in your notes packet, which we're on page 7 with this chart here. We're just looking at the first part of the chart to begin with. So don't write too big because I do want you to add little teeny tiny pictures in there. You can kind of see, like on mine, little sketches there. So do your best to kind of sketch something in here with us. So from the pictures from before, okay, what antigens, what team jersey does A have? It has A. That's the easy part. Okay, what antibody, and then B, has B. AB has A and B, and O has neither or none. So I want you to write those in, but also draw it. Draw a little sketch. Okay, you don't necessarily have to do like circles and triangles or whatever. You could use just 2 different color pens, whatever works for you. But do try to depict it in some way. All right. When I wrote mine, I literally just wrote little A's on the outside and little B's and little AB's on the outside to show mine. And then again, O has neither. If you need to pause the video, you sure can. I'm going to keep going. So the antibodies present are going to be opposite, like we just saw on the sheet. So the antibodies are called anti-B because, again, we're like against B's. No B's allowed if you have A blood type. And so it's going to fight or attack those. If you had B blood type, it would be I'm against anything with A's on it, anti-A. ABs have none. So no anti-A or anti-B. So don't draw anything here. And then for the O blood type, it's going to have both anti-A and anti-B on there. So O is going to attack anything that's not O because it's got both. A won't be attacking anything because it doesn't have any antibodies. So who can donate to who becomes slightly confusing. But if you have antigens straight, antigen is me, my jersey. Okay, what am I? Antibodies is who I'm against. Okay, that's who I don't. That's who I want to fight. Okay, that helps us know who can donate blood. So you should have seen from our lab simulation that A can give blood to both A and AB because it does not have any of the markers that would fight A. Basically, anyone who doesn't have anti-A can receive A. So when you think about who the blood can be donated to, we look at the antigen in this scenario, meaning they're getting the whole red blood cells from an individual in a red blood cell donation. And so these guys are who they need. We want to make sure that their body's not going to attack it. So they can't have any anti-A's present, okay? Which means, okay, they can only donate to A and B, or sorry, A and AB because of the antibodies present. Now, who they can receive blood from is slightly different because instead of looking at the antigens, when we talk about whose blood I can take into my body, I got to pay attention to what antibodies are present in my body. So if you're A blood type, your blood can go to A people or AB people, but you can't receive AB blood because you have anti-B antibodies. If we put this in you, then those antibodies would attack it. So instead, we look at what antibodies are present when we figure out who you can receive blood. So if anyone has a B on them, they can't take it from them. So the only people they can receive blood from in this case is A and O. If you want to, this is a great point to pause the video. This is where in class I would ask you all to help me fill out the rest of the chart here and see if you can figure it out using this logic. So maybe pause, see if you can fill out the chart, and then go ahead and resume and see if you were right. All right, did you pause? Who knows? Now, before we go through the rest of these, the thing that the antibodies do, again, is cause agglutination. Let's make sure and write that word somewhere down on your notes because antibodies cause the clumping, that agglutination, during a transfusion if they are the wrong blood type. This is, again, why A can just get A and O. So hopefully you wrote down what you thought, what blood types can be given to who here. So if you are B blood type, we want to look at what antigens you have to figure out who you can give your blood to. Somebody that matches. Well, B can give to B. B can also give to AB because, look, they match. All right. But they can receive blood. We need to look at what antibodies they have. Well, they are against anything A. So they can't receive this one. Yeah, they can take B. They cannot receive AB, but they can take O. So they can receive B and O blood. All right, now AB blood, again, we're going to look at who they can donate to. In the case of AB blood, we want to again look and see who they match with. Well, they match with A, B only because they have both. So ABs only can give their blood to other ABs. However, just because you have a short list of who you can donate to, actually, they can receive blood from, check this out, no anti-A's, no anti-B's. They're not going to fight any blood type at all. This is why they can take everybody's blood. They can take A, they can take B, they can take AB, they can take O, which makes them the universal recipient. Okay. Now, O, sort of the opposite. Okay, so again, who can they donate blood to? Well, we look at what antigens they have. They have none. That means they can give to everybody, making them the universal donors. However, again, when I look at who can they receive blood from, I need to look at what antibodies are present in O blood. Well, O blood has both. So they can only receive from other O's. So here's sort of the good news, bad news in this scenario is that AB blood is the most rare. But good news if you have AB blood is that you are also the universal recipient. So even though your blood type is rare, you could, if you were in need of blood, you could successfully take somebody else's blood. O, who can only get one type of blood type, other O's, they are doing okay because O is the most common blood type in every ethnicity. And so there's never, I shouldn't say there's never, but there is rarely a shortage of O blood type specifically. Now, the second piece that we need to know when we are donating blood or receiving blood is what your RH factor is. And the RH factor in the video, the supplemental video I have you watch today after this, is the world's most dangerous blood type. It's very fascinating. It's a great thing for you guys to watch, and I hope you do. But they discuss the fact that blood cells have actually lots of factors on them. There's lots of things that are on our cells identifying them, but we are mostly concerned about A, and then if they're positive or negative. Those seem to be the two most important when we are receiving and giving blood. So if somebody is Rh positive, what that means is that their cells have the D antigen. They don't call it the Rh antigen, they just call it the Rh factor, but it's a different type of antigen present. So if they're positive, they have that antigen. I put Rh down here. D's not wrong. You won't see that on the test though. But Rh positive means you have the antigen. That's all. Rh negative, much like it sounds, it does not have it. Now there's only one antibody for this antigen. So if you are Rh positive, you do not have the Rh antibodies. So there's no antibodies that affect it. This makes positive a little bit more accepting of other things, whereas the Rh negative has none as far as the markers go. And if it has antibodies, they would be against that D antigen, or they'd be Rh positive antigens. And there's an asterisk there. I'm going to explain that here in a minute because it's not true all the time, or depending at what stage of your life you're in. However, who can donate to who in this scenario? Okay. Rh positives can give to other Rh positives. They can also receive blood from both blood types because they don't have any antibodies. They're not going to fight anything. So I'm O positive. I could receive O negative blood, just A-OK, no problem there. And I could receive O positive, the exact same type as me. Now, if I was O negative, that's the universal donor. Okay, there's no markers on the outside of those cells. So they can give their blood to a positive person like me. However, if they are negative, they can only receive negative blood because they do have the antibodies. And I wish there was a picture here that showed some antibodies floating around on the outside here. But because they do have antibodies, they would attack any positives in their bloodstream. Okay, so again, if you were AB, the universal recipient, but you were AB negative, one of the rarest blood types, you would need to receive negative blood, whatever kind it is, would need to be negative. So let's talk about what I mean by this idea that there are sometimes maybe Rh antibodies. You don't necessarily need to write all of this down, but it only develops if you've previously come in contact with Rh positive blood. So kind of like the chicken pox. If I've never had the vaccine for the chicken pox and I've never actually had the chicken pox, then I won't have any chicken pox antibodies. Um, and so my body wouldn't recognize it the first time it came in contact with it. The same is true with Rh negative blood. They don't have any antibodies, but they will create them if they ever meet any blood that is positive. If you got the wrong transfusion and you were negative, you'd be okay. But if you ever happened to get positive blood again, you would not be okay. But the big problem is in pregnant women. So if a mother is Rh negative, doesn't matter what other blood type she is, but if she's Rh negative, and I have a little picture here, okay, you would have to have a positive father in order to have a positive baby. And if the baby is positive, much like the mother is, and they don't normally test the blood of babies. If you did amniocentesis, you might know your baby's blood type, but it's not very, common. But if they know that the mom is negative, they're going to take some steps to prevent this. But if they don't, what would happen is in the first pregnancy with a positive baby, between the placenta and the mom's bloodstream, there's some crossover. So some of that baby's positive blood would come in contact with the mother's negative blood type. Her body would recognize that as an invader. And her body, the mom, the negative mom, would start to build those Rh antibodies in her body. However, it will not attack the first baby she's born, that she gives birth to. That's positive. So here's a mom who had a positive baby, and she developed antibodies for any positive future offspring. So the problem is if the mom gets pregnant again, okay, and again, if it's the same man and he's positive, the chances of the baby being positive is also increased. If the second baby is positive and she has these antibodies present in her body will attack it. And then you would have a miscarriage. So not on the first, but on the second child that's born. So to prevent this from happening, if any of you ladies have a negative blood type, you will receive some injections during your pregnancy that will keep your body from developing these Rh antibodies to begin with. So you can have a positive baby, no problem. You won't get to this step. And then we wouldn't get to this step where miscarriage would happen. So anyways, if in future life you end up being one of those individuals that has to get that shot when you're pregnant, it's just because you don't have antibodies yet, but you could develop them for your offspring. But if mom and dad are both negative, there's no risk of having a positive baby. It's genetically inherited. So negatives and negatives just make negatives. But negative is a little bit more rare. Speaking of, I want you to, underneath this table, okay, write the following, okay, so that we know for sure. Again, it's on the table, but that I know you know what's the universal recipient, universal donor, most common, and then the last one that's being covered up says least common, at least in the US. Okay. So the universal recipient we know is AB, specifically AB positives. Positives can take anybody's blood. Universal donor is O negative. They have no markers on their cells, so they can give to anybody. The most common in the US is O positive, like yours truly. And then the most rare is AB negative. And so that's the most rare type. And AB negatives need to receive, and for sure, negative blood, but they could receive somebody else's blood type that was negative if needed. O negative is a great supplement there. All right. This is a very confusing topic, I know. So if you need to go back and watch this or look at the slideshows posted, or the slides that I went over with you today, by all means, go back. This shows the same information we've already gone over, but I do like this is it says frequency of each blood type in the US population. And depending on your ethnicity, it also varies as well. In general, though, O is the most common, then A, then we have B, and then AB is the rarest. So if you have B or AB blood, you're rare bird. All right. So the other piece that you're going to see on this assignment is how we determine blood types. So if we wanted to figure out if somebody was A or B, what we would do is we would take a small little drop of their blood, like a little finger stick, and then treat it with some serum. Now, anti-A serum, this is basically serum filled with anti-A antibodies. Right? If they clump, that means that it's A blood type. So these are the antibodies, the things that make the clumping, the attackers. Right? They're antibodies. They're against that body. They're going to fight it. So if clumping occurs, right, that tells you, okay, that's the blood type. And so we have the same serum here that's B, and if clumping occurs. And so this is your homework assignment, is to figure out what blood type they have. So I won't show you these. Just kidding, I will. Don't look at those. You can 100% rewind and just see those answers. But instead, I wanted you guys to watch, fill out these notes. These are on page 2 of your packet. Now, black and white does not do it justice as far as showing you these colors, although the red and the blue is not super important. But at the very top, I want you to write anti-A and then anti-B, so that you can figure out what blood type each is. So when we have clumping like this is what it looks like. That's agglutination. And that's just clumping of the red blood cells as a reaction with the anti-A, the antibody serum. It's not my body. And so we can try to determine, we have all 4 blood types represented here, and we can figure out what blood type they are based on the clumping that's occurred. So here, both of them clumped. It means that they have A antigens and B antigens because they clumped in the anti-A and anti-B serum both. So this would be type AB. You can just write AB to the side. You don't have to write all those details. Type A, okay, because it clumped here and not here, it didn't react with the anti-Bs, but it did clump on the anti-As. That tells me that it's only A blood type. Same down here. It only clumped with the B, agglutination, and so it must be a B blood type. And at the very bottom, no clumping occurred, which means there must be no markers, no antigens, and that would be an O blood type. Now, to figure out if they were positive or negative, we would need a third serum here that was the Rh factor. And if it clumped with the Rh anti-serum, that means that it's a positive. If it doesn't clump, then it's a negative. But we would need the third test here in order to identify what blood type they officially are. All right, that is the very end of our notes here. Thank you for a long day talking about blood types, and do watch that most dangerous blood type video. It's very fascinating. And then complete your homework assignment for today. Have a good one.